An action potential is an electrical charge that travels along the axon of a neuron. This nerve impulse carries information that needs to be relayed to other neurons or effector cells. A synapse is a site of transmission of electric nerve impulses between two neurons, or between a neuron and a gland, or muscle cell. Synapses between the axon endings of one neuron and the dendrites of other neurons are called axodendritic synapses. Those between the axon endings of one neuron and the cell bodies of other neurons are called axosomatic synapses. There are two types of synapses, electrical synapses and chemical synapses. Electrical synapses consist of gap junctions that intimately connect the cytoplasm of adjacent neurons, allowing ions and small molecules to flow directly from one neuron to the next. Neurons joined in this way are said to be electrically coupled, and transmission across these synapses is very rapid. Chemical synapses are specialized for the release and reception of chemical neurotransmitters. Although close to each other, presynaptic and postsynaptic membranes are always separated by the synaptic cleft, a fluid-filled space approximately 30 to 50 nanometers wide. Now let's look into the process of the synapse. First, a nerve impulse arrives at the synaptic end bulb of the presynaptic axon and opens the voltage-gated calcium channels. Then, the increase in calcium triggers exocytosis of synaptic vesicles and the release of neurotransmitter into the synaptic cleft. The neurotransmitter crosses the synaptic cleft and binds to the receptors on the postsynaptic neuron. The postsynaptic membrane permeability changes, causing an excitatory or inhibitory effect. When the potential reaches the threshold, it triggers one or more nerve impulses. The neurotransmitter receptors mediate changes in the membrane potential according to the amount of neurotransmitter released, and the duration the neurotransmitter is bound to the receptor. There are two types of postsynaptic potentials, EPSP or excitatory postsynaptic potentials, and IPSP or inhibitory postsynaptic potentials. EPSPs are graded potentials that can initiate an action potential in an axon. At excitatory synapses, neurotransmitter binding causes depolarization of the postsynaptic membrane. Each EPSP lasts for a few milliseconds, and then the membrane returns to its resting potential. The only function of EPSPs is to help trigger an action potential distally at the axon hillock of the postsynaptic neuron. Neurotransmitter binding to a receptor at inhibitory synapses causes the membrane to become more permeable to potassium and chloride ions. This leaves the charge of the inner surface negative and reduces the postsynaptic neuron's ability to produce an action potential. A single EPSP cannot induce an action potential. EPSPs must summate temporally or spatially to induce an action potential. In temporal summation, the presynaptic neuron transmits impulses in rapid-fire order, and in spatial summation, the postsynaptic neuron is stimulated by a large number of terminals at the same time. IPSPs can also summate with EPSPs, canceling each other out. Synaptic delay is the rate-limiting step of neural transmission. Neural transmission across the chemical synapse is comparatively slow compared to the speed of an impulse. Neurotransmitters must be released, diffuse across the synapse, and bind to receptors, causing a synaptic delay of 0.3 to 5 milliseconds. Neurotransmitter bound to a postsynaptic neuron produces a continuous postsynaptic effect. It blocks the reception of additional messages and must be removed from its receptor. The removal of neurotransmitters occurs when they are degraded by enzymes, reabsorbed by astrocytes or the presynaptic terminal, or diffuse from the synaptic cleft. If you like this video, please hit the like button below, and subscribe to my channel to see more videos in future. Thank you for watching. Bye.